A little brief introduction of Dr. Mohanty. He is a distinguished medical professional with multifaceted career in healthcare. He currently serves as a program director and pan-India head at the Center for Biorepository and Biobanking at HCG Cancer Hospitals in Bangalore. In this capacity, he oversees the collection, storage, and management of biological samples critical for cancer research and personalized medicine. Dr. Mohanty holds an MBBS MD in general medicine and DM in cardiology. He has received several accolades and including the International Fellowship of the European Society of Cardiology in 2020 and multiple gold medals for academic excellence. Beyond his clinical and administrative role, Dr. Mohanty has a strong academic background. He has engaged in research at the School of Oriental and African Studies, focusing on intersections of design, digital culture, healthcare, and entrepreneurship. His doctoral research examines the future of healthcare through digital technologies with comparative studies between India and the UK. Dr. Brojan Singh has not joined us now. So about Professor R.K. Tiwari, who is another chairperson for this session. He is an associate professor in the Department of Physics at Hindustan College of Science and Technology in Mathura, India. He holds an MSc, MPhil, and PhD in physics. With over 22 years of experience in academics, industry, and research, his research interests includes nanocomposites, optical properties via absorption spectra and IR, charge storage and transport properties, thermally stimulated effects in polymers, dielectric properties, and thermoelectric effect. He has supervised several PhDs, BTech, MTech, MPhil students. Dr. Tiwari has published numerous research papers in journals and conferences, and he has also been involved in organizing the national and international conferences. So, is Dr. R.K. Brojan Singh there to join as a session chairperson? I don't think he's there. So I think he's not there. So we can start with the session. So I would request my persons to kindly take the charge. Online.
Online. YouTube तो बोलिए पहले स्टार्ट करिए कहाँ पे स्क्रीन पे लिखा
Good morning, everyone. So it's my honor to invite our first speaker of this session, Dr. Amit Yadav. He is a principal scientist at Translation Health Science and Technology Institute, HCST, in Faridabad, India. He earned his PhD in biotechnology with a focus on computational biology from the CSI Institute of Genomics and Integrative Biology, IGIB, in Delhi. During his doctoral studies, he developed MassViz, India's first peptide identification algorithm for mass spec data and proteome analysis, an integrated analysis suite recognized by MIT TR35 award in 2013. His research mainly focuses on PTM, post-translational modifications, and big data in translational health sciences. And his work encompasses protein-protein interactions, biological networks, computational analysis in metabolic diseases such as non-alcoholic fatty liver disease and cardiovascular diseases. He has received several prestigious grants, including DBT IYVA, DBT Big Data Initiative, SERB Supra, and DBT National Network Project. And he's also the elected Executive Council member of Proteomics Institute of India. So next, I welcome Dr. Amit to take over the podium and deliver his talk.
A very good morning to all of you, and thank you for the kind introduction. Am I audible?
Please hold for 2 minutes.
Okay. Am I audible?
Yes, sir. You can carry on, sir.
Okay. Thank you for the kind introduction, and I thank BBIF India and Dr. Virendra to have me here. I'm going to talk about advancing personalized medicine through proteomics, the role of biobanks and data repositories. Since I work in the big data field, we generally keep abreast with all the data repositories, specifically in mass spec and proteomics field, and how we can leverage that for personalized medicine and how we can integrate genomics and proteomics data. I also believe that with the advent of biobanks, we will begin to have better research outcomes even in India, like we have in the Western countries. With this, how do I move my slides? Okay. So this is the institute where I work. This is THSTI in Faridabad. And if you get some time, please visit there. And before I start my talk, let me acknowledge the superheroes who do all the work while I just get to talk. So these are the people who run the show. These are some of the areas that my lab is currently working in, but I will not focus or harp on it too much. I will specifically talk about liver mod forms and proteoforms. So if you forget everything from my talk, please remember this word proteoforms, and I will believe that I would have done a decent enough job so that you can remember this. So we all know that a gene can form different types of mRNA because of alternate splicing, and what we call as isoforms are formed, and they slightly have sometimes slightly different functions. What we tend to forget is that there are also a lot of post-translational modifications like glycosylation, phosphorylation, acetylation, methylation, ubiquitination, because of which these proteins can actually take different kind of forms, and all these various forms that they can form, they may be structural or functional, and we call them as proteoforms. The important thing is that in disease biology, we generally tend to only measure the proteins, but we do not measure whether the glycoform is up or phosphoform is down. And I'll show you with examples why this is important. One thing missing from this image is mutations. So when we talk about single nucleotide polymorphisms that come from GWAS studies and talk about disease propensities, these mutations, if they are non-synonymous, can also result in amino acid change in the peptide, which we generally tend to call as a variant peptide. So these are also part of the proteoforms. So this is the basic of mass spectrometry. So how do we measure all these proteins at large scale? So we take a complex disease sample, we digest it using an enzyme called trypsin, which will cleave these into peptides. After creating this complex sample, because if your sample had thousands of proteins, now you have probably millions of peptides. You don't need to send them at one time to the mass spec, because otherwise all you see is noise. So you need to separate them so that they enter the mass spec one at a time, and therefore you will be able to measure or sequence them. Mass spec is a chemical weighing balance that measures mass to charge ratio of an analyte, but it can only measure if they get some charge. Therefore, all these peptides are protonated, separated through the LC, and when they enter the mass spec, you see somewhat excuse me, you see something like this, which is called as an spectrum. So the peaks in yellow are actually coming from these peptides. You may also have some background noise. The mass spec is told. So what we tend to generally do these days is called tandem mass spec. So in tandem mass spec, you have one mass analyzer after the other. The one is used for discerning the mass to charge ratio of peptides. The other one is used for sequencing these peptides. So these top intensity peptides are selected and sent for second round of mass spec, which is called as tandem MS. So in tandem MS, these amino acid bonds are broken down, and you get something like a, so again, something like this kind of a spectra where you do not know which part comes from which sequence of amino acid, but you get their mass by charge ratios. So if I have a model peptide like this here, and I break them down randomly, just like it happens in collision-induced dissociation or the second round of mass spec, since I know the mass of each of these residues, it is very easy to calculate their specific weights, molecular weights. And therefore, using these specific molecular weights, what we create is shown on the right side as a theoretical MS-MS spectrum. So all the intensities are same. Since this is not coming from the experiment, you see all the intensities are actually same. So using this reference theoretical library, you can match your experimental data over the top, and then based on how many peaks are matching, you can actually sequence the peptides. I have shown some in red color because if there is a mutation, the T might change to some other residue, and therefore there will be a mass shift, which we can account for while calculating the theoretical spectrum. This can also account for post-translational modifications. For example, if I have a threonine phosphorylation, all of these red masses would increase by a 80 Dalton shift. And you can also tell from this that a normal peptide or a non-modified peptide will not accurately match to its modified spectra. Therefore, you need to have modified theoretical spectra to match. With this is just a common shotgun proteomics workflow. This is important to understand what I'm going to talk about later. So you have a sample on one hand. On the computer, you have a FASTA database which contains all the protein sequences of the given organism, human, for example. You generate peptides. Similarly, on the computer side, you perform an enzymatic digestion in silico and you create peptide library from the genome of interest or proteome of interest. In the mass spec, you separate and ionize these and generate millions and millions of MSMS spectra, each probably arising from a peptide. On the other hand, you filter the candidate peptides. So now let's say I want to interpret this MSMS spectrum. I say that there is a parent mass of 2000 Daltons. I look in this database and say, bring me all the peptides that have mass somewhere around 2000 Daltons given the instrument error. And all these peptides are selected as candidate peptides. For all these candidate peptides, we can create theoretical spectra, and they will all be sequentially matched with this MSMS spectrum to find the best match. So these are things that are automated and done on a computer through what is called as a match scorer or the peptide identification algorithm. So there are several peptide identification algorithms, but we'll not go into the details. So now the important thing is when we identify mass spectra, we generally tend to identify only 1/3 of the spectra. The other goes into the dustbin. So there are several reasons for this, but I'll focus on two important parts. One is when we do not know which post-translational modifications are occurring in the data, then these get identified. And if the peptide is not present in the database, for example, mutated peptides, and if there are some non-canonical peptides not yet identified in the genome or not yet identified as expressed from the genome, so we will not be identifying them. So the first part is covered, the PTMs are covered through what is called as a blind PTM search, and novel proteins or peptides can be identified through integration of proteomics and genomics data. So this is just a complete picture using genome and transcriptome information, integrating it with MSMS spectra from proteome. What we can identify are variant peptides. We can find novel genes if they are being expressed. We can find peptides if they are referring to some novel exons or novel splice junctions. Sometimes even pseudogenes get translated, and we can use all of this information, alternate start or alternate end sites for gene model correction. So this way, proteogenomics comes to the rescue and combines genomics and proteomics data. When we try to identify mutations, similar way, if I have DNA or RNA sequencing data, we know mutations at genomic level. So using 6 or 3 frame translation, depending on whether it is coming from DNA or RNA, we can create a customized protein database. This is not part of general shotgun proteomics because these databases are 100 times bigger than the normal databases, and therefore very compute heavy. Not all labs would try to do this. So therefore, if you create a customized protein database, you can again use the same algorithm and try to match these spectra to find mutated peptides. Now there are some challenges there. So just as one case example, we took 21 different regions of human brain, and we took healthy human brain data sets. These are millions and millions of spectra in terabytes, and we downloaded from public database, and we wanted to create a reference mutation proteoform database for human brain so that we can use these in disease studies. But this is not easy. So we have to create a computational pipeline. In this pipeline, we first identify the raw data against a reference proteome or uniprot proteome for that matter. So we have around 40 to 50,000 proteins there. After this, whatever spectra remain unassigned, we fetch out those. And now we try to search against all novel kinds of proteins that are not covered in the reference proteome. So this is, as you can see, 1.6 million translated protein sequences. So these are all predicted sequences. But when we actually match the spectra to peptides, now we know that these are actually occurring. And then once we still have a unassigned leftover spectra, now we create this custom mutation database for all the dbSNP SNPs known. And using these mutated peptides, we can find what is called as variant proteoforms or variant peptides. So through this, we try to create a reference human brain atlas for variant peptides. But we also identified that variant peptides are generally identified wrongly, because due to these large databases there are a lot of false positive. So we did a simulation experiment and created a tool. This tool looks at all the output from a proteogenomics experiment, and whatever has been identified, it again puts it to the test, but in more detail. What does it do? So we have created single variant peptides, double variant peptides. We also kept wild type peptides, and also peptides that are unrepresented in the data. And so in the database, we did not keep wild type sequences to match, but we generated their spectra. Similarly, we created unrelated peptides in the database, but we did not create any spectra for that. Through this simulation, we know if single variant matches to single variant of the database, it is correct. Double variant matches to double variant peptide, it is correct. The rest, all of them are false positive. Using this stringent metric, we evaluated this algorithm, which was reusing all the mass spec information present in the spectra, like intensity, continuity of the ions, specific comparison with the wild type peptides, whether the variant is scoring better or the wild type is scoring better. We could identify most of the correct ones as confident or semi-confident. And so this data, we performed several other comparisons with published data, and we found all the important things that were correctly identified. And we could see that roughly 65% of the spectra that variants in a general proteogenomics search are actually false. So using this, we understood that we need to remove a lot of false mutations, because otherwise we'll be groping in the dark and we'll waste our resources on things that should not be tried in wet lab for confirmation. So that's what we tried. After this, we did this in the complete data sets that we have downloaded, and we found that roughly 5 to 10% of all peptides, even in healthy human brain, were variant peptides. And this is one of the examples. So the top one is known, the bottom two are not known, and we found them also in the same data. So we compiled all this information in a database called HubSpot, wherein if people work on Alzheimer's, neurodisorders, they can, if they make a discovery, they can go back to this database and try to find if their variant proteoform is already reported or not, because measuring proteins is now not enough. As a second example, we work on non-alcoholic fatty liver disease, wherein if you have 5% fat in the liver cells, this is called steatosis. If you also have inflammation and ballooning, it will be called NASH. After NASH, you may progress to cirrhosis with fibrosis and hepatocellular carcinoma. From these three stages, it is very difficult to come back only through diet and exercise, and there are no biomarkers except for a biopsy, which is difficult to do. And roughly 1/4 to 1/3 of population all over the world is suffering from this disease. There's only one drug, and the side effects are also not known. So we created a PTM mapping pipeline called ModQuant, through which we performed large-scale searches for modified peptides. And we are trying to create a liver proteoform atlas. Using a tool called Modest, we divided this chunk into different cores of a computer, so that just like a bank queue model, the job can become smaller. Otherwise, it takes 100 times more compute power and also time. So we thought that let it take time, but at least it should be able to run on a decent enough computer. That's why we called it Modest. And we know that when we try to search for modifications without a priori knowing which modifications are there in the data, you tend to make a lot of false discoveries again. So we created two more tools called PTM annotator and mod locator, which will correct the site localization using again the ions from the data, and also annotate which modification it is. Otherwise, the tools only tell you that it is 80 Daltons. You have to infer that it is a phosphorylation. So we showed that the log score we have created works well in a public standard data, and PTM annotator is also able to 98% accurately identify the modifications. The rest are because they are isobaric in nature, so we cannot distinguish. So we took a public data set of 6.18 million spectra from a cohort of NAFLD patients, and we tried to search for some modifications using our ModQuant algorithm. We identified that whatever is the non-modified part, which is already reported in literature, roughly equivalent number of modified peptides and proteins were also found. In fact, modified proteins were higher in number. So we said we should look at this in more detail. So we had looked at phosphorylation, methylation, and acetylation. And we showed that this is an important thing. If you forget everything else, you must remember this one. Generally, we say that albumin is going up or down in disease, but since albumin is 95 to 99% of human plasma, any disease you want to measure, the ratio will not change too much because the other proteins are very low in abundance. So what we observed here, that if we look for mod forms specifically, we see that different isoforms or mod forms of the same protein which are supposedly doing same function do not behave same in the disease conditions. You can see the ones that I have circled here. So you can see one isoform of albumin is showing a different pattern from two other isoforms of albumin. So this tells you that whether it is albumin, apoM, or apoC proteins, which are liver transporters, so they behave differently in the different stages of this disease, and it is important to measure these things. Otherwise, we would actually miss out on a lot of hidden biology only and only because we are fixated too much on the protein, because protein is just a bulk of everything combined together. So this summation does not help the clinical picture. So we created a map of these mod forms across different disease conditions, and each dot here represents a mod form instead of a protein, and the circle size represents what abundance it was present in the sample. And therefore you can see a similar protein can be annotated at 2 3 different places, but you can be rest assured these are either phosphomethyl or non-modified forms of the same protein. Therefore they are shown like this. And this tells us that we should look at proteoforms and mod forms in more detail rather than having a cursory look. And with this, we identified several PTM signatures. This is just a curated list. The list is much bigger. And now, working with AIMS, we are trying to validate this in independent cohorts. And that's where, apart from databases, biobanks will come into picture. And using different public data sets, we again showed that around 50 to 70% of proteins are modified in different data sets, even if we use different proteins. So modifications are everywhere. So measuring proteins might be simple, but it may be wrong if we talk about clinical accuracy. And for precision medicine, we need to go towards more complex answers, although we hope that they are better, or at least explain the scenario much better way than what we were able to do before. And as with all these big data kind of projects, it's easier to talk with a cleaned up data, but usually we are always drowned in huge data piles. So thank you. I think I might have slightly exceeded my time by a minute or two, but thank you for your patience. And uh, it takes a village to do anything worthwhile, and a lot of people and a lot of grants have made this possible. And I express my sincere gratitude to Dr. Birendra Yadav, BBIF organizers, and all my students and collaborators and funders that I'm able to talk about all this here. Thank you so much. So over to the chairs. If we have some questions and if they allow, maybe I can take a few. Hello.
proteomic marker.
So,
Sensitive marker, because there is a different level of data set between a brain tissue proteome versus the fatty acid liver disease proteome. So what is the how do you decide upon the threshold of the power of the study?
okay, so maybe because of time constraint, I was not clear enough. So these were two different studies. They had no connection in particular, first of all. Hello? Hello, am I audible? Am I audible now? Okay. Yeah. So first of all, these two studies are completely independent of each other. They were parts of different PhD projects. So I just talked about these in two different contexts. The first was, hello. So the first was on brain mutations, and the second one was on liver mod forms. So the second one has a cohort from German dataset. The first one was from just a compilation of different. First one was just a compilation of 20-30 different data sets. So there was no required power of disease because we are not calling it a biomarker. It is just a reference atlas. It's a qualitative data set. The second one is a quantitative data set where we have everything calculated. So this is already a published cohort from German data. And we are also trying to validate it in the cohort from AIMS. Hello.
Yeah, thanks, Amit. Top, I would like to invite Mohammad Farooq, Senior Principal Scientist at IGID. Is a researcher at the CSIR Institute of Genomics and Integrative Biology with an impressive academic background. His MBBS PhD. His research focuses on molecular biology, genetics, and neuroscience, with a particular interest in the molecular genetics of hereditary ATXs. Dr. Farooq is currently involved in the projects such as GOMED, genomics, and other omics tool for enabling medical decision, and Guardian Genomics for understanding rare disease India Alliance Network. His expertise span neurologistic, cerebral ataxia, neurodegradation, human genetics, movement disorder, human molecular genetics, clinical and adult neurology, and demyelinating diseases. So hello, good morning. So thank you, BBIF. Thank you, Dr. Yadav, and thank you, Dr. R.K. Tiwari, for
the nice introduction. So I'm going to talk about something about, like, say, let's say the purpose of biobanking for genomic research, and also some experience from my own lab, my own institute about, like, say, creating a resource that we call now as biorepository of a lot of DNA samples, cellular models, etc. So to begin with, I would like to say like this is the basic framework for like biobanking for genomic research, and where we look for the structured sample collections, archival, and also the associated research data generation. And it has a very important component of clinical material, that is what we call as clinical annotation, clinical features, or clinical information. So in a typical biobank, mostly it pertains to healthy subjects in typical sense what we usually gather, but they may have different phenotypic traits as well associated with them, depending upon the original structure of the biobank. So in biobank, so the purpose, the researcher look upon the biobank, the take home, or maybe what you call the transaction. If we have to understand, that is basically to understand the genome information in the form of allele frequencies and genotype frequencies. So and the genotype per se. And overall, the purpose is to understand all this information, the allele frequency and the genotype for doing population studies, population genomics, as well as the to make this useful for the medical genetics research. So technically, when we talk about this population genomics, we are interested in understanding the diversity of population, the migration history, the linkage pattern, the imputation, the haplotypes, IBD scores, that is identity by descent, and then the foremost application of the biobank data for GWAS studies. And the foremost immediate application of biobank data is for day-to-day clinical activities for variant interpretation that we often encounter, that patients who are referred for genetic diagnosis, we identify genetic variations and we have to compare it with the reference data sets, and that is what we compare it with the biobank data. So it's very important for molecular diagnostics. Carrier frequency estimates, it can give definitely. Risk prediction, it helps. And then establishment of polygenic risk score. This is just general about this thing, and this is the current. So with the advent of this next generation technologies, even not only sequencing, but all the high throughput technologies, they coincide with this idea of biobank development. So these are different biobank and their repository, the strength, the number of samples, etc. So we have very popular UK biobank. And following that, we have also the National Biobank of Korea, which is a very large biobank, but that basically deals more with the patient, and versus the UK biobank data that deals with the general population. These are the general, the sort of the data sets and the tissue or the sample resource they have. So these are, like, say, mostly they have the genotyping arrays, whole genome sequencing, RNA sequencing, methylation data. Then also they look for the collection of blood samples, DNA, RNA, and so forth. So overall, the advantage, like, say, the biobank data from genomics point of view. So we look upon this allele frequencies and etc. for medical genetic application. So why it is important? Suppose in this in our context, in Indian context, we usually have less what you call a presence in global data sets. So that you can see that mostly they are Europe-centric data sets, and we have fairly minimal, you can say, representation of South Asians. And among South Asian, we do not have India represented as whole in the all the global data set. So what is the importance of adding more samples, meaning more data sets? Why these are not sufficient? Because, like, say, every genome or every DNA brings its own unique variations, and more you add the samples, more genomic variations catalog you increase, and that is how you facilitate your variant interpretation. So likewise, these are the general the statistics, like you have in this all that genomic repertoire of variations. You see most of the variations are unique variations, singleton. They are less recurring, and confined to sometimes the individual country or maybe individual community. And also helps this, all the genomic resource helps in understanding the diversity. It is a typical PCA plot for understanding that one population is not yet the same with the other population at genetic level. We do have similarities, we do have dissimilarities as well. And this also helps, meaning if you have the genome level data available with your biobank, having the leads from the Jiva studies, you can really do a fine mapping sort of a stuff. And these are, like, say, I'm just trying to tell you that the purpose of the biobank data for doing genomic research. This is what I thought to give a glimpse of that. And this is how, like, say, this can facilitate all that genome information that is coming through our own established sort of a biobank that has come, is going to be sort of accessible and available, that is from Genome India team. And so I think a lot of data has been generated, a lot of trade phenotype information is there, and we are going to have a nationwide, maybe say you can say democratized access to that data resource. So now I'm just going to talk about the sort of research that we have been doing in my lab, and that's where I'm going to talk about that importance of DNA and cell biorepository. I call it biorepository, not biobank typically, because that is mostly specific to a single center, but we are open for collaborations, etc. But mostly it pertains to a disorder that is a hereditary ataxias. And hereditary ataxias is a genetic form of cerebellar ataxia. So where the cerebellum, like among neurodegenerative disorder, so you have different cell types and different brain regions, and the specific phenotype in the form of, say, one type of neurodegenerative disorder as Alzheimer, hippocampus, Parkinson disease, you have motor neuron disease. Likewise, we have cerebellar ataxias, where the involvement of cerebellum is seen, the cerebellum degenerate. This is overall the journey for us. So how the modus operandi in last almost close to 3 decades was that, like, we ran a sort of a pan-India sort of a network, and centered at main tertiary referral center, All India Institute of Medical Science. So every patient who visited the clinic at AIIMS, that was a specific unique clinic for this particular disorder, ataxia clinic. So our basic modus operandi was high quality service, then derive research out of it, and then that culminates into translation. So technically, it's a high quality service in the form of mutation screening. So every subject weekly visits or daily visits at the AIIMS, at AIIMS neurology department, comes to us for genetic diagnostics. And once the case has been diagnosed, the diagnosis report goes back to the patient. The sample is added to our resource bank, and that we utilize for future research. And with that, like, say, we try to address a lot of questions like with the mutation screening, what are the prevalent mutation type, what are the hotspot mutations, and from their research part, we have been able to identify, we have been able to study the origin of mutations and identify novel mutation. We are able to successfully make machine cell line models, and as well, whatever the knowledge we generated, we were able to successfully commercialize with our own panel that we have come up with this disorder. And this is sort of a like one aspect of genetic spectrum of different ataxias. If I have to cover, so far in last 3 decades, we have around 8,500 like DNA samples with us from all the subjects that were collected through All India Institute of Medical Science. We have detailed clinical records available with us, and what we see even before the advent of these NGS technologies. That 6 mutations, 6 genes, and its mutation could explain 30% of our cohort, meaning 30% of subjects of ataxias were found positive for certain mutations, and 60% remain undiagnosed. And what are these 6 mutations? They are sort of non-conventional mutations. Generally, when we talk about mutations, we think through in terms of point mutation, substitution, something like that. But here, these 6 mutations are tandem nucleotide repeat expansion, like we see that in Huntington disease. So likewise, for ataxia disease is majorly caused by the CAG repeat expansion in different genes, and those genes are 6 as indicated by different disorder they cause. So technically, for that here I would emphasize that these repeats are in general important for neurodegenerative disorders. They might be the major driver of the pathologies rather than the conventional mutation, but not that repeat alone is the culprit. So further, we went on to characterize this large uncharacterized cohort after we have screened the 6 mutation, where we can look upon. Coming back to the conventional thought process, single nucleotide variations. So we have done some whole exome sequencing of 600, close to 620 subjects, and this is the repertoire of different genetic subtypes of ataxias that we see. So we see autosomal recessive ataxias by not the triplet repeat, but the single nucleotide variations. 46% were autosomal recessive, 31% were dominant, and 22% were in the genes, which may either manifest as an autosomal dominant fashion or autosomal recessive fashion. So this is what the genetic spectrum of our cases among the uncharacterized category. And also, like beyond that, even after deploying whole exome sequencing, we were not able to completely characterize our uncharacterized cohort. Still, 30% remain unsolved. And again, we look upon, as I said, the repeats are important for neurodegenerative disorder. We are again looking for more genes where repeat may be the cause. And by keeping that the huge resource in hand, so there was a discovery last year. NEJM reported that this FGF14 gene earlier, which was known to cause SCA27, and the mutation mechanism was point mutation. Now, last year, we a group from Henry Holden's lab, they reported that the same gene with the GA repeat expansion in the intronic region causes SCA27B disease. So it's sort of the same sort of ataxia type, but the mutation mechanism are different. One same gene, one where the point mutation, and the other instance the repeat expansion. The interesting aspect is the motive. Again, that GA repeat was already known to cause ataxias in other gene that we call frataxin. So what I'm trying to imply here is the motive type is important and its length in the gene. So if that motif is placed in any other gene, that may still cause similar sort of a neurodegenerative manifestation. And this SC27 we have screened in our entire cohort, and we were able to identify close to 23 patients. And what did we do after identification of these 23 patients? We looked upon the origin of mutation, that when did we get this mutation in our country, sort of. Maybe it's a de novo, or is it an ancient one? So we calculated the age of the mutation, and we were able to find that almost like it occurred, it came into the country almost 22,000 years ago. So what it tells now, so meaning it's such an old variation mutation, we may have reservoir of this the mutation in our population, and we may see more prevalence of this particular type in Indian context. So that is what this finally the conclusion was from this. Then the other thing, we are having learned things like genetics from ataxias, even that I'm pursuing since my PhD time. So we delve upon these other genetic disorder. Whatever we have learned from ataxias can be apply on the other genetic disorder. When I say apply, means the modus operandi, meaning high quality service and develop resource, do research, and then translate. So likewise, we have we initiated this concept of GOMET, that is a patient-centric research-based diagnostic service, and 2015 we launched it, and with our clinical network we started getting referrals, and over a period of almost like 5-7 years, we were able to get 14,000 referrals from various department, various diseases. And not only neurology, but pediatric onset, typical all the genetic disorder which we usually encounter, thalassemia to rare of rarest. And so sort of we were able to make panels and sort of understand the diagnostic spectrum. We created cohorts. We made the databases of genetic variation that we have identified from this GOMET, the repository, and we sort of made it available to the, we made it as an open access through this ClinDB database. And so we also estimated the different diagnostic yield from our GOMET repository for different neurodegenerative disorders, and a good amount of diagnostic yield we were able to see even disorders like muscular dystrophy, 60% is using our panels that we have developed through GOMED. And then hereditary spastic paraplegia, 50% we were able to diagnose, and so on. So overall, the approach, like what we finally aim for actually. So technically is that like all this mutation screening lead to the identification of patient, the diagnosis. So what? So we can make them as a group and make them as we can actually recruit them in our future in our next study setting that we call as natural history cohort. So that you once patient has been identified, then longitudinally you can study year by year and collect the data and understand the disease history. So what happens? Then they are the trial ready cohort. So what happens now? From all our the repository, patients have been binned into different disease categories, and now we are part of international registries as well, and where the trial ready cohorts are the aim. So likewise, we were successful in setting one international registry for one ataxia type, Friedreich ataxia. That's a one, and with that. That is helping us now. This trial ready cohort is helping us having a discussion for a trial from Biogen in India. So this will help us connecting our patient to global science, and as well the patients after this diagnostics to this treatment possibilities. There is a chance for this disease inclusion in rare disease policy of India, because there is a framework, or maybe there is a need that the disease should meet the following criteria, then only it can be considered for rare disease or policy document, so that patient can get the benefits. So, for the same disease, like say we also had some cell line resource as a model from patients, and that helped us in making collaboration with Wisconsin University, Dr. Azim Ansari. And there they had identified one synthetic molecule that can restore all the deficiency of Friedreich ataxias, and that was a science paper. And how we participated in this, we helped them in validating their molecules testing in our patient cell line models, in primary cell line model from this Friedreich ataxia patient. So overall, I'm just trying to tell you that from patient to diagnostic to cell line models, and now coming to the possibility of therapeutics development. And similarly, one of the rare other disease that other than ataxia that we have encountered through our routine day-to-day GOMED activities. So one rare disease, intractable seizure, ceroid lipofuscinosis, and there it's an intractable seizure causing disease, and there the problem is this loss of function of the CLN5 gene, and the gene therapy, which is a trial is happening in UK, and so we connected our patient to this particular trial, and the patient is in queue, apparently not yet included, but this gives us an opportunity, meaning connecting, at least if we do not have things sort of readily available in our country, at least our diagnostics effort or our cohorts can help connect our patient directly to the whatever trials or therapeutics available in the world. And still, but we are not yet dependent entirely on all the efforts happening in abroad. We are also developing our own capacity. We have made almost like 100 patient cell lines from different types of diseases, not only like ataxia. These are made since I worked on ataxia, so we have a lot of these ataxia cell lines, but we have like say DMD, Duchenne muscular dystrophy cell lines. We have rare motor neuron disease cases cell lines, genetically proven motor neuron diseases. We have also rare genetic diseases. Even the new disorder we have identified, that is skeletal dysplasia. So all these are available in the form of this lymphoblastoid cell line, and we are open for collaboration. We also make iPSCs. So of these 100 cell lines, we have made 30 iPSC lines, so that you can derive them into your required cell types. Meaning, if you are working on neuronal disease, you can convert your iPSCs into neuronal lineage, and you study further. And so these are just some examples that not only the mechanism and what you call that therapeutics development, they also help us in what you call validating the pathogenicity of a variation, because all the time when we deploy a genetic test, it doesn't tell that okay, like I found a variation and that is pathogenic. At times the variation is of unknown significance, we do not understand. So this patient cell line also helps us in directly validating that particular mutation in the same person's cell line. So we don't have to engineer any like a mutation into another secondary cell line. We have the cell line from the same subject. What you need to do is just you have to identify the correct assay, perform the assay, which can be the readout of your genetic defect. So here one example I would say, this was like mitofusin Z. So this because of this mutation, mitochondria will not divide. So as you can see here, this patient, so these are like say typical, it's a neuronal problem. You can see mitochondria is fragmented form, and in mutated line, the mitochondria are more round shaped and sort of having a sort of a rounded appearance. So, so these are like, say, as I said, like LCL line, we have been making iPSC line, also converting them into iPSCs, and that allow us to collaborate and also use them possibly for therapeutics development. We have a lot of these different rare diseases, ataxias, and then we have P62 mutant cell line from patient, and also the critical ataxia I said, and these are like say available even global, like for open for world as well. We collaborate and we participate in the drug development process. And this is that what I said in the beginning, like say all our genomics effort will be available from biobank perspective. The Genome India database will host all the frequencies of the genetic variation that we have sort of estimated, and that this will be readily available for the access, and that is what we participate. And with that, I would like to say thank you for the opportunity, and that's it.
Hello, yeah. Thank you, Farid, for the lovely, exciting talk. And there's a lot of work which shows that the biorepositories can be a hub for genetic mutations, and we know the translation, how quick is quite essential there for mutation screening. Any question, a quick question from the audience? Okay. I just have a quick question, curious one. So you have a phenotype-driven mutations which you are reporting, right, for mutation screening, right? Then you have acquired mutations which actually come in the course of your investigations. How do you now report those de novo mutations which are come in the acquired in the form of acquired mutations which could possibly be linked to the phenotype, which we discover later? And do you deposit those mutations in the ClinVar as a pathogenic mutations leading to the phenotype?
So technically, let's say we don't talk about this acquired mutation. Acquired mutation we may say sort of observe when we make the cell lines. So when the cell lines passage during culture time, they may acquire new variations, but technically the impact of them are not that something which will detriment the cellular health. But we, as cell passage more and more, we encounter a lot of deletions and duplication in them. Otherwise, this repository is just like the static DNA sort of a sample, and anytime, like suppose you have a new discovery, you go back to your data set.
But is there any correlation of those mutations which you see in the cell lines to the pathological phenotype?
Sort of not. Sort of. No, we have not seen that.
Yeah. Thank you, Dr. Farooq. Dr. Farooq, just it is a fantastic presentation. I like to know that there is one biobank coming in your institute. One biobank is coming in your institute. Yeah. So how you are going to use the biosamples from that biobank in research, and whether that biobank, what is the source of the biosamples for that biobank?
So technically, as I said, like say this, whatever I mentioned about the research which I do, we call them as biorepository for particular research, particular that thing. And biobank typically, when we talk about UK Biobank, and for that matter, it's the same thing which we are talking about Genome India cohort. So basically, we are like say aiming at characterizing, right, or like say all the phenotypic information, all their health indices, all their biochemical parameters, and all that is coming from healthy subjects, self-declared healthy subjects. So they are like say not so exactly same, but yes, they definitely provide the baseline health of all the evolutionary health history.
Okay, I think in the interest of time, we'll have to end the session here. Thank you, Farid, for the exciting talk. And we'll go ahead with the next speaker. Our next speaker is Dr. Anita Mahadevan. I think everybody knows her. She's a leading neuropathologist at National Institute of Mental Health and Neurosciences, NIMHANS, Bangalore, specializing in neuroinfections, peripheral neuropathies, epilepsy, developmental neurobiology, and brain banking. She has held key positions including professor and associate professor in the Department of Neuropathology at NIMHANS, and she previously served as the senior scientific officer at Human Brain Tissue Repository, the Brain Bank, and has conducted extensive research in neuropathological disorders. And Dr. Madhavan has authored 283 publications, including 253 journal articles, 13 reviews with over 3,000 citations, and an NH index of 24. Her contributions to neuroscience and brain banking have been widely recognized. She has played a critical role in advancing neuropathology research and demands, and has significantly contributed to human brain tissue repository, supporting studies on the neurological disorders. And I'd just like to update that she's also the PBIF Pioneer Award for this year. Big hand for her. Thank you.
Thank you, Dr. Abhishek, for that very generous introduction. And thanks, Dr. Virendra, for the invitation to be here, and thanks BBIA for that award, which I don't know if I really deserve it. So what I'm going to do today is just take you through, I know the last 2 days you've been listening to all kinds of biosamples, but perhaps what has not been spoken about is what I think is the most valuable resource of all, which is an autopsy. I'm going to concentrate only on what are high-risk autopsies that we have done over the last 3 decades, and whether it was really worth it is the question. So you may ask, what is a high-risk autopsy? I will just quickly go through what is, by definition, a high-risk autopsy, our experience, and whether it was worth it. I think to define a high-risk autopsy is kind of redundant now, because everybody relates it to COVID. And I think COVID really brought this into the forefront of value of an autopsy, which I'm really happy about, because not happy that we had the pandemic, but one of the good things it gave us is it told us the importance of autopsy. So every little information you got during the COVID pandemic, be it how the virus enters, be it what it does to every organ, what receptor it uses, and all of this information which finally went to make a vaccine was thanks to this group of courageous pathologists in New Orleans who did the very first autopsies in COVID at a time when vaccination was not there. Okay? So keep that in mind. Of course, the rest of the world tried to do all kinds of things at this time to do the autopsy. They came up with this kind of indigenous way of protecting themselves when they are doing autopsy, etc., etc. But at that point of time, ICMR did give us guidelines on what we should do to do autopsies during this pandemic and to establish a national biorepository. But much before COVID came up, I think we have buried many of various neurological infections. I'm sticking to neuroinfections because that's what comes to our institute. Of course, several of these also affect other organs. So a high-risk autopsy is applied to any autopsy which is related to those which have a high risk of transmission to those who are doing the autopsy. Now, the Advisory Committee on Dangerous Pathogens has classified this into 4 groups based on the risk of infection, the likelihood of spread, and access to treatment or prophylaxis. So group 1 is very good, doesn't lead to any diseases. So all of the spinocerebellar ataxia, etc., which Farooq was talking about, I can do without any kind of protection. Group 2 may lead to disease and risk to employees, but it doesn't spread to other humans, and there is prophylaxis or treatment available. Group 3 is what leads to severe disease. There can be a significant risk to employees. It can spread, and prophylaxis may or may not be accessible. Group 4, of course, is classically Ebola. Don't do anything. Don't go near. Okay. So I just looked at CAP guidelines when COVID broke out, and interestingly, this is what they said. So high risk group 2 is all of these. Group 3 is what we get a lot of in neurological practice, as in rabies, you have dengue, anthrax, TB, and the prion diseases. These are, they still say, manageable. But group 4, interestingly, they say, don't get involved in these. And autopsies are effectively banned in the UK. So this is essentially Lassa, Ebola, etc. So what about our own data? When we looked at our own data of autopsies that we did, right, in our own practice, we found that almost 44% die of CNS infections. I know the whole world is so enamored by cancer, so enamored by neurodegenerative diseases, they don't seem to want to pay attention to a huge bulk, which is infections in several countries. Okay? And when we looked at our own engineering controls that we had at that point of time in our autopsy, we have a beautiful autopsy suite. It is really clean. It is so clean that our residents always say that it is cleaner than the mess and they would rather have their lunch here. We also have a separate viewing room, a dissection room, an office, etc., which is what is recommended. Now, what is the precautions that we use for every autopsy? That is irrespective of what the clinical diagnosis is. We assume that it is infectious. So what we do is use routine PPE. I think everybody is very, very familiar with that, which is just double gloves, protect your eyes, use a patient, use a mask, a plastic gown and apron. And we also have a necropsy saw, which is connected to a vacuum suction to make sure that it doesn't cause aerosols. So that is what that autopsy room has contributed to most of what we store in our brain bank. So it was started in 1995 by Dr. Shankar, and we are trying to carry that same legacy forward. 3 decades have gone by, so you may ask us, what do we have archived? So again, it is infections, which constitute almost, you can read that, it's 59% of what we store. So if you look at our scorecard of the high-risk group 3, we've done HIV during the pandemic. We crossed 190. Neurotuberculosis, as one of my very close neurology colleagues says, by virtue of being born an Indian, tuberculosis will be there. So we had a huge group of tuberculosis. We've done rabies, prion, COVID-19 too. I've done 19 autopsies when we still did not have the vaccination, 2 anthrax and 2 dengue. Anthrax because we didn't even know it was anthrax. But I'm still standing in front of you here and I'm fine. And no autopsy acquired infection, touch wood, in any of those attenders who assisted me. And I must tell you, those attenders are the ones who told me to start COVID-19. They told me, madam, you've done so many other things. Why not COVID-19? We are there, let's do it. And that's how we did it. Okay. So we look at how useful this has been. When we were in the middle of that epidemic of HIV in our country, lots of things came out thanks to all of the autopsies. One is that HIV clade C is the most common in India, and that had enormous repercussions on the vaccine. We also knew that clade C, interestingly, has a completely different spectrum of HIV-associated infections. If you looked at our country, we always had opportunistic infection as the most common, whereas the West would say that HIV-associated dementia was most common. And the reason they would say we are not having dementia is because all our patients die early, 1. 2, our clinicians are not able to diagnose dementia, both of which we do not agree with. So when we looked at our autopsy series, we realized that clade C, interestingly, does not predispose to dementia. It is a completely different genotype, and the way it behaves is different. And this was, we could establish that thanks to Dr. Ranga Uday, who was at GNCSR, because we had a lot of stored, fresh, frozen autopsy samples. We also realized that in those patients who survive after successful treatment of opportunistic infection, the question we asked was, will they develop because they live long enough? The answer, interestingly, was again no, as long as it is a subtype C. So a lot of important epoch-making publications came out of this, which had biological significance. We also helped to validate indigenous diagnostic kits. Sorry, indigenous. We had the HIV kit of NACO, which is now used all over India, validated at our bank. We had the Cysty Circus kit. We now have a multiplex PCR, which can detect 21 different pathogens, all in 1 drop of CSF. Now, a lot of our companies are coming out with indigenous kits. Who is going to validate, and how are they going to get these confirmed, clinically characterized cases? That can happen only if we bank our samples. So all of us are looking for that magic biomarker. We had lots of omics studies that was just presented. We're looking at an actionable molecule. All of us want precise diagnosis. We want a prognostic. We want a predictive marker. So you may ask, what have we done with the omics era? I need to tell you that most of the omics I don't understand. Comics still remains my most favorite. But we did get into that. We jumped into that. And all of the samples that we had banked in our brain bank, we had given it to the Institute of Bioinformatics, and that is Akhilesh Pandey's group, who realized it's such a huge gold mine. And they jumped in and they did a lot of proteomics, which I will just give you a taste of what samples contributed to literature. So this was a quantitative proteomic study that we took from all of our tuberculous meningitis patients whose samples we had archived. There were several novel markers of significance. We had this chitinase 3 like one going up 16-fold. He's nodding, Farooq. I know why he's nodding. Interestingly, at the same time, there was omics being done for glioblastoma. And there too, Chitinase 3 was up 21-fold. If you look at neurodegeneration, I'm sure you'll find the same thing up. We realize that is just a host response. It's microglia that are producing it. So don't get excited. So like that, we had lots of stuff that had come up. We need to understand whether it is host or pathogen, which is throwing that up. But what was interesting to us at that point of time were those which were down-regulated. Renin, which is important for cognitive function, interestingly, was down-regulated. So were several other protein transporters, again, which have a role in cognitive function, which tells you that if our patients are going to survive opportunistic infections, I'm not talking about HIV related, I'm talking about good old tuberculosis, they may be predisposed to neurodegeneration earlier. So we need to find biomarkers which can predict cognitive dysfunction in survivors, and we are trying to validate this on a cohort which we are following up. Now, we also did, of course, the transcriptomic studies. We validated it with immunohistochemistry on brain sections, and that's amphiphysin, which we know is a synaptic vesicle protein which was upregulated. We did the same thing across all other infections. So this is picture-perfect cryptococcal meningitis, which is a very gelatinous exudate on the surface. It was the second most common fungal infection after tuberculosis in the HIV cohort. We also had toxoplasma encephalitis, which was the most common focal brain lesion in HIV. And interestingly, there was a lot of overlap when we looked at the data. So it again tells us that we need to segregate which is host-mediated, pathogen-mediated before we jump and say this is prognostic or diagnostic. Another interesting virus that we could look at because of the large number of autopsies was rabies. You will tell me that so much is known about rabies, but interestingly, there are several things which are not known. We still don't know why that dramatic hydrophobia occurs. Never occurs in animal models of rabies. We still don't know why you have an incubation period which can be 5 days to 6 years. So if I get bitten by a dog on JNU today, 15 days from now, if I'm still alive, it doesn't mean I should be happy. 5 years from now, if I'm still alive, I should be happy. So we don't know why that variable incubation. What we do know, however, is 100% fatal. So do you know why it kills? Interestingly, we don't even know that, but we do know it kills. So we looked at a few of these as to why this incubation period is so variable. We looked at 3 groups of patients that we had. One was a very short incubation of up to 2 months, another up to 1 year, and another longer than that. We realized that inflammation increases, no surprise in that. These are the Negri bodies. I don't know if you can see it, but you have to believe me, the arrows are pointing to beautiful Negri bodies. When you look at immunohistochemistry, that's the amount of viral antigen. The pattern varied with increasing incubation period, it starts to compact. No big deal there. But then we did a painstaking neuroanatomical distribution across the entire human brain. And wherever the brown is where the antigen is. I won't take a class in anatomy, but this is a beautiful neuronal marker where every cranial nerve nuclei is standing up. But if you map it, where the red is where maximum antigen is, where the blue is where it is least. So if you look at the very short incubation period, everything is localized to the midline. Midline is the worst because those are where your critical areas are. So if that gets involved, certain death. But if you take the long incubation period, entire brain gets involved. And interestingly, the last to be involved is the midline reticular activating system. So that's just to wake you up. If you take a long route to reach this convention center, you will be in time for the valedictory. If you take the shortest route, you'll be in time for your session. Okay, so that's what the rabies virus does. Depending on that, your incubation varies. But the $1 million question is, why do patients die? There is no edema in the brain. The neurons which are affected just don't die. So why do patients die? We still don't know the answer. We tried to look at apoptosis because everybody at that time was speaking about apoptosis during viral infections. Interestingly, we did not, there was a lot of viral antigen, but no apoptosis in the brain. The only thing showing apoptosis, interestingly, were inflammatory cells. So that is a brilliant strategy if you think about for the virus. What it's doing is killing off all immune cells so that it can survive. Just one flaw in its strategy, the host is dying, and we still don't know why. But if you look at animal models, lot of apoptosis. Just to tell you that human and animal, you really cannot equate. You need to use human tissues if you want to find answers. Did proteomics give an answer? Interestingly, yes. Again, we like the down-regulated. Apoptosis was down-regulated. Immune mediators were down-regulated. All of the synaptic proteins and neuronal markers were down-regulated, telling you that It is not a structural damage of cells, but it's probably a neuronal dysfunction that is killing. We looked at a lot of CJD that we had done, and there was a progression in the pattern of deposits of prion protein, and this just added to literature. So basically, we are looking at an actionable molecule. It could be diagnostic, it could be predictive, it could be prognostic, but the bottom line is that you need to look at host versus bugs contribution and not just jump at the markers that are thrown up at you. So if you want to find a cure, you want to find a vaccine, the most critical step, I think, is you need to have well-characterized banked tissues and fluids to support research. Otherwise, it's, I think the famous saying now is GIGO, is that right? Garbage in, garbage out. So that's, we need good, well-characterized samples so that all neuroscience research, science research is supported in this country. But just having samples is not good enough. You have to talk, the clinician and the basic scientists have to talk to each other to make sure that it is clinically relevant research. So I'll just end by telling you that autopsies, and I love to keep saying this, still contributes enormously to science, and we need to continue to do autopsies. For that, you just don't need pathologists to be there, you need clinicians to understand the importance of it. And I think our own scriptures say Mrityorma Amritam Gama. So that's where we are headed probably, and we know death is certain. So I would say, let us donate, let us start. Now, I want to end by thanking Professor Shankar, who started this brain bank, and we're just continuing that legacy. This group of mortuary staff who are with me day and night and always willing to do autopsies. Without them, I cannot do anything. And our entire brain bank staff who are thankfully growing in numbers. And to that, I think I would like to thank the funding agencies, particularly ICMR, who has really believed in us. They helped me start a network of brain banks across the country. So we have satellite banks now in AIMS, Bhubaneswar, as well as PGI MER. And we have now, thanks to ICMR, started a national biorepository where we are storing all of these infectious disease samples. But just banking is not good enough. We need users, and that is what I'm trying to enthuse this group here. So please use it. Otherwise, all that effort that has been put in to bank it will be in vain. Thank you very much.
Yeah. Thank you, Dr. Nita, for this intriguing delivery of how important is the role of repository in understanding the molecular neurobiology of neuroinfections. I'm sure you agree that without the structure and systematic archival of these autopsies, which is the first of its kind in India, we look forward to an era of not a multi-omic biomarker, but actually a disease-specific, infection-specific marker which will come up, or a bug-specific marker, right? Exactly. Yeah, open for questions. Yeah.
Hello, ma'am. It was very interesting talk, and I came to know a lot of things which I was not aware of it. So, and my question is, like, when the repository is there for autopsy samples, so the first criteria is the patient or the body which is has consent for this thing, right?
Absolutely.
The other thing, like, what are the steps, like when the brain is removed or the particular brain sample is taken out through autopsy? So, what is the time period in which it has to be frozen or stored? Second question is like very highly pathogenic cases like HIV, as you were telling. In that case, the autopsy sample is safe for long-term storage. Like it will not disturb the other or infect the other people.
Yeah,
this is my question.
Thank you for that question. I'll take the last question first. So the reason I presented this is just to tell you that we need not fear doing high-risk autopsy. If you take PPE, regular, you don't require even your negative pressure air ventilation systems, etc. You can do it safely as long as you've got your personal protective equipment in place. Okay. Second thing, you asked me how long, what should be the post-mortem interval? Up to 24 hours is good enough to do all of their omics data. In fact, we did one study where we looked at the best marker, I think, mitochondria. We looked at the oxidative markers, and we were surprised ourselves that up to 12 hours, you can still do mitochondrial studies. If you can do that, you can do any other kind of study. So it of course, it depends on your downstream events. If you're looking at RNA, after 12 hours, we found that it degraded, but up to 12 hours, even that is preserved. If it is protein, up to 24 hours is not a problem. DNA, you can take it out of Egyptian mummies, means you can take it out of frozen samples. Okay? So ideal, less than 12 hours. Up to 24 hours, it is still good. You asked me whether it's infective or not. So up to even a week, people have been able to isolate HIV from frozen tissues. Up to 10 years, people have been able to take out Mycobacterium tuberculosis from good old formalin fixed tissues. Okay? That still should not scare you. As long as you take protection, you can safely do any kind of stuff. Now, you also need to remember that most of the viruses, they need a host. They're all obligate intracellular. So the minute you take it out, its infectivity is gone. You can safely handle it. In fact, they say that within 8 seconds, if you take out HIV virus, it is no longer infected. It needs a host. Okay? As long as you don't cut yourself. If it is an inhalation agent, as long as you wear an N95.
Another part of that when the sample is taken out.
Yes.
So what are the you categorize your samples where you are keeping it or storing it? Like HIV sample, this is the catalog, this is the place where this kind of highly infectious sample is stored.
Yes.
So that the person who is taking care of that whole place where the biobanking is done, so that person is aware that this is a high risk area where the sample is highly infected.
Yes, we do. But at the same time, all our technical staff, attenders, everybody are trained to assume every sample is infected. If you tell them that this is not infected, they will not take the same precautions that they would do when they would handle an infected sample. So dictum, assume everything is infected. But yes, we do store it separately. We have one freezer for prion, one for HIV. But then we also run out of space. We can't have one for every infective disease. Yeah. But then we do separate out infective versus non-infective.
How many years you store it? From guidelines?
1995. Still being used. So I don't believe that it is only useful for 10 years. We have had from 1995, whatever samples we've been preserving is still continued to be used. Good for moleculars, excellent for proteomics. The only this thing is RNA degrades. I'm happy you asked so many questions.
Okay, I think we'll have to close the session. Thank you, Dr. Anita, for this enumerating talk. Thank you.
In continuation to the series of talk, our next speaker I would call Dr. Devajyoti Chakravarti, Principal Scientist, CSIR. Dr. Chakravarti, Principal Scientist at CSIR, Institute of Genomics and Integrative Biology, has recently achieved significant milestone in the field of CRI-SPR technology as he co-led the development of Tata CRI-SPR test, the world's first diagnostic test to deploy a specially adapted CAS9 protein for detecting SARS-CoV-2. In 2020, he was honored with the CSIR Young Scientist Award and selected as one of the 30 EMBO Young Investigators, recognizing his outstanding contributions to life sciences. So, welcome to Dr. Devajyoti Chakravarti.
Thank you. Thank you, the organizers, for inviting me. So, in a statistical graph, you have those dots which are outside the bar. We call them as outliers. I'm a kind of an outlier because I'm not going to talk about biobanking. I don't do much biobanking. And also, the work that I'm going to talk today about is also not going to be so much about RNA. So it will be more about gene editing therapies. But I'll try to do justice to the topic here as I discuss them with you. My work actually comes from the point of view of how to make gene editing therapies possible here in India, and what are the hurdles and challenges to overcome that while we do that in the process. Okay, so I come from an institute which already, thanks to Farooq, you have an idea that it's one of the first, if not the first, Indian institute to do the sequencing of the Indian genome, human genome, and also it was the institute which later on went on to do sequencing of a lot of other neighboring countries' genomes as well, like Malaysia, Bhutan, and so on. So once this genome sequences has been done, I think some of us who were hired later on were told that you have all the information. Now you also know what are the mutations which cause these diseases. Could you possibly go and do some gene correction and see if this is possible to cure these, to develop therapies based on these gene sequences? And just to tell you that, gene therapy using gene editing, I don't have to explain to this audience here what gene editing means, is not a science fiction anymore. And at this point of time, there are people who are just like us, who are, in this world, whose genes have been altered and their diseases have been potentially cured. So it's definitely, something that is very real. But unfortunately, like this statement here in Nature, which is, I have just highlighted it in red over there, as you see here, that these therapies, although they are available, it's not going to be available to people in a lot of countries, including India, very soon. I can already tell you that. The first FDA-approved sickle cell therapy, which came in December 2023, Casgevy, or even Lyfgenia, which is the lentiviral therapy, is priced at 20 crores per individual. So it's a one-time shot which will cure your disease, but you'll never be able to pay for it. So no point even thinking about it. But the interesting part is that the largest number of patients for these diseases, whether it's sickle or HAV, is actually from Africa and India, where these people do not have access to these kind of therapies. So what I'm going to tell you today about is that, one, we have to isolate the problem as it is, why it is so, and what can be done and is being done here to actually break that barrier of making such therapies affordable. So it's great to have high science and fantastic discoveries, even a Nobel Prize, but it doesn't make any sense that these will be out of reach for people who actually need it. One of the reasons that it has not reached people is actually, if you follow the patent war for CRISPR, and you know that there has been a long tussle on it, and there are two major players, Jennifer Doudna and Emmanuel Charpentier, who won the Nobel Prize, and Feng Zhang, who did not win the Nobel Prize from MIT, but won the CRISPR patent. So therefore, there has been a long series of litigations that have happened over the last almost 10 years on the courts, and it is still ongoing till 2022. And these are just to tell you that, Berkeley even argued with lab notebooks as to who discovered CRISPR first and all. But the point was that in the end, Broad MIT got the license or the patent for CRISPR, which means that any kind of activity using CRISPR has to be licensed from the University of Harvard. Now, that's okay. We know in biology, almost big discoveries are always patented, and the way to come out of it is that you license these technologies. That would have been nice had been followed the same way. But if you look closely at how CRISPR is licensed and gene editing is licensed from Broad Institute, it forms a very, very complicated process, because it says that we have identified the diseases that has to be worked on. We have our subsidiary companies who work on them. And if you want to develop something, you have to go through them. So today in India, if, for example, as ma'am has said, that there is a new disease which has been identified and a gene therapy is possible, you simply cannot go and start using CRISPR and treat patients with it. You would first have to approach Broad, find if there is a potential partner that Broad has already identified, work through them. Alternatively, you pay a license fee of close to 150 million US dollars. So no Indian company, no company in Africa, no company in any of the places which do not have that kind of a bandwidth would come and make a CRISPR therapy. This is the background for the cost of CRISPR. That's why a drug costs 3 million US dollars or 20 crores INR for a single dose. And that is the same reason when, if you think of a disease which is affecting patients who are the poorest, such as sickle cell anemia, for example, which happens in African populations or in tribal belts of India. There is no way that any kind of a company in good faith will come and say that we are investing it into a completely lost case altogether, where there is no way to recover the cost. So if I just kind of summarize what I have told you, there are four major reasons which I can tell you about. One is, of course, the drug itself, gene editing itself. You're trying to actually do a gene correction in the human genome of 3.8 billion letters. So the safety aspect is paramount. You have to work on the safety. Pricing is oftentimes determined by how you source these reagents. So a lot of, and this, there is a good example is the CAR-T field, where you have seen a lot of companies coming in the Indian space. The majority of reagents, when imported, when there is no indigenization, has a very high price tag. So that's another important point to think about. IP I have already covered. And of course, awareness, whether our doctors and our patients are aware that such kind of novel genetic therapies exist and whether they are going to be a party to it in case that kind of a therapy becomes available. I will cover each of these very, very briefly throughout my talk. So our story starts in 2019 when me and my colleague, and most of the work that I show here is actually done with Dr. Shofik Maiti, who is the current director of IGIB and also nucleic acid biophysicist, who comes in with his DNA protein interrogation expertise, and I come with the molecular biology expertise. We figured out in 2019 that there was an alternate or let's say an orthogonal Cas protein from a bacteria called Francisella novicida, which has got a tremendous high specificity of DNA recognition. And if you look at how it interrogates with DNA, if you take a DNA piece, and I hope all of you are aware of how CRISPR works. It's a protein which requires an RNA to go and bind to DNA, double-stranded DNA, opens up the DNA, cuts the DNA. Now, if you give a substrate of DNA and you give a substrate of DNA with mismatches, these are in the genome, these are called as off targets basically. Then whereas the most widely used Cas9, which is almost used across 99% of the labs around the world, Streptococcus pyogenes Cas9, would actually cut your off target to the same extent as the wild type Cas9. What we found out, this Cas9, which was completely uncharacterized by that time, called Francisella novicida Cas9, was only cutting the target DNA but not cutting the off target, as we call it. Okay. So that told us that, wow, that's very remarkable, because inside a test tube, if this Cas is able to distinguish between what is a target and an off target, then imagine if you give it inside the cell where you flood the cell with proteins, you can actually have a very, very precise Cas, which will only go to that one letter in the DNA out of 3.8 billion letters. That was our hope, and very specifically make a change and correct a disorder. That was what we thought. But first of all, we needed to know how this was all happening. How is it able to distinguish? And we, to cut a long story short, we did a lot of biophysical experiments to see how this protein and DNA was interrogating. And don't go into the numbers, just to explain to you, this is called a microscale thermophoresis experiment, which measures the binding affinity of protein to DNA. And as we found out that this Cas9, when it incorporates or when it interacts with the target DNA versus when it interacts with the target DNA, which has got a single mismatch and single nucleotide polymorphism, is not binding at all. And that's why there is this shift of binding affinity to almost micromolar range. Now that was very, very interesting, because that told us that the Streptococcus pyogenes was actually bound to its target and off target with almost the same level of affinity. But FN-Cas9 was not so, which means that if you could give this protein a piece of DNA and you give this protein another piece of DNA, which has got just a single mismatch, this protein can tell you which of the DNA has that mismatch, right? Simply because it will not bind to it. And if you can think around it a little bit carefully, you can almost use it as a type of diagnostic of a single nucleotide polymorphism without the need for DNA sequencing, because the protein is telling you that either it binds or not binds. So that essentially led to what I was introduced by, so which is called Feluda. Feluda was a diagnostic technology which we developed during COVID-19 times, and is also the world's first commercially approved paper strip CRISPR test that came from India. Essentially, what it does is that it tries to detect DNA sequences without the need for any kind of sequencing methodology. Now, it also got the regulatory approval. Now, how it works is in principle what I just told you, is that we kind of married this CRISPR biology with the paper strip chemistry. We have made some modifications and converted this all into almost like a pregnancy strip, where you have a band which shows whether there is a positive signal or not. And the name was, of course, because we were two Bengalis, and that point of time, Feluda is, of course, a name of a Bengali character from Satyajit Ray novels. I don't see any Bengalis in the audience, probably. Otherwise, there was always a some kind of a discussion about it. But nevertheless, doesn't matter. So it comes. The full form is FN-Cas9 editor link uniform detection assay. We kept the names. Our competitors from MIT and Berkeley kept it as Sherlock and Detector. So that's why we kept the name. And of course, the idea was that, you at that point of time, you were going to field, to the field in villages, and we saw that we could detect things such as even sickle cell anemia, which is a point mismatch using this kind of a CRISPR technology without the need for sequencing. You don't have to really send these samples, wait for a month sometimes, because, in this particular case, the nearest sample center was CCMB Hyderabad. And by the time they would send their Sanger sequencing results, the patient would be lost. So that was all what we were trying to do at that point of time. But of course, when COVID came, the government said, stop everything else. You just have to work on COVID. So we thought that, okay, then we can actually try and make this into a test for COVID. And then, just you cannot make a test in the lab, doesn't make any sense. So we had to find our partner. So Tata came in. Tata, of course, had been only selling steel and salt by then. But they said that, okay, we also want to get into diagnostics. They made a new company called Tata Medical and Diagnostics. They converted this whole thing into a kind of a, how to say, robotic setup, put it in, I don't know if it's yeah, they converted this into a robotic setup, put it behind a truck, made robotic pipettes of this CRISPR components, had a manufacturing pipeline there, and essentially they called it Tata MD check. They said that this was very useful for going into places where qPCR machines were not available during the pandemic, such as in Northeast. Now that's all fine. You can write papers, you can publish things. You have so much of media attention and all. But that actually shifts the entire focus from the real work, which was that if you have a cast like this, you don't want to make diagnostic kits out of it. The real application for this would be to actually correct genetic disorders. That's what we started off with. Now, like I tell you that it's not just for COVID-19. Of course, Feluda is applicable across the spectrum. This work recently got published with Dr. Govind Makharia at AIMS, using Feluda for detection of clarithromycin-resistant varieties of bacteria in peptic ulcer samples. Or, for example, this work which we are currently doing in the Northeast with in Arunachal and in Assam, where rotavirus pandemic, rotavirus is a huge issue there, and from stool samples, very early detection of rotavirus. But anyway, so what I wanted to tell you is that the actual application for gene editing, and if there is, very highly precise gene editors, would be in treating genetic disorders, and we should not shy away from trying that. One of the issues that we had was that this Cas9 from Francisella novicida was very specific. I already showed you that. But it was also not very efficient inside cells in terms that, it was giving us a gene editing ability, which was way, way lower than that of Streptococcus pyogenes Cas9. Now, one way to increase the efficiency we figured out was actually to do protein engineering. So, to cut a long story short, we collaborated with scientists at the University of Tokyo. And what we were able to do was actually engineer this protein with almost 50 different types of mutations in them, and test them to find out that we could identify what we now call as engineered FN-Cas9 proteins, EN-FN-Cas9 proteins, which can actually perform even better than Streptococcus pyogenes Cas9 with having the same level of specificity. So point mismatch specificity across the genome in terms of editing the genome. And it can also work across multiple cell lines, multiple loci, and most importantly, when you compare it with other high fidelity Cas9s which have been developed recently, it actually outperforms them. What was very startling was now if you go across the genome and you look for off targets, this is just for sickle cell anemia. What we saw was that some of the Cas9s which have entered clinical trials have entered so with almost close to 1400 different types of off targets or places in the genome where it can potentially cut. The Cas9s that we have engineered over here, we call as engineered Cases, which do not have any kind of detectable off targeting. If you take some of these off targets that we get from digenome sec and we validate them in patient cells as well, we do not see off targets. So that gave us a very, very specific Cas9, but more importantly, it gave us a kind of a novelty in terms of engineering. I'll tell you where that becomes very important. Of course, Other thing that we made was that we could make second generation, third generation CRISPR effectors such as base editing. And base editing essentially is a method by which you put the Cas9 on a single site in the DNA, and then you can make a single base change. And just to show you, this works very beautifully. This was work done with Dr. Indu at LV Prasad Eye Institute, where a patient was lost vision, is a blind patient, has a point mutation at the RPE65 gene causing Leber's congenital amaurosa. And if you can use these engineered base editors to actually restore the protein function complete, which has been lost from childhood, back in the eye organoids, when you make eye organoids on mini eyes on the dish. And that's very beautiful because it gets 100% gene correction. And this is something we are taking ahead for a clinical trial at LV Prasad Eye Institute. So all this was necessary and important because that led us to the next step, which is to the possibility to generate a what we call in this field as FTO, or freedom to operate, only when we could have a US patent granted. So this patent came last year for engineered FNCAS, the first Indian patent to actually have a CRISPR, to have the first Indian CRISPR patent. And its impact was so much that the government actually invited us to Kartavya during as a guest. And as we have seen this always in the TV where the MiG-21s fly over you, and I always thought that how beautiful that would be to once get an opportunity. I never realized science would give that opportunity to actually go and witness this right there as a guest. Now that's all fine. Again, you show this, you get the patent. What next? So the idea of actually developing therapies for Indian patients comes from the fact that these kind of therapies also have to be developed from scratch using India-centric modalities. You simply cannot take what works in the West and push it over here and think that it will work out all in the end. That doesn't work. Sickle cell disease in India happens predominantly in tribal patients. Now think about doing a gene therapy for tribal patients where even hydroxyurea is not available at all centers. And these patients live with a monthly income of sometimes a few hundred rupees. To explain to them what a gene therapy is, I thought was a very, very complicated process, let alone do it to them. But then when we go to the villages of Chhattisgarh or Nagpur and all these places, and then you have farmers coming and say that, look, Dr. Saab, there is an ulcer in the leg, and I know that there's a cure which is called CRISPR. Can you tell me whether you can do something about it? That's when I told that I understood that awareness point which I mentioned in the first slide is not something we need to fight with. Awareness is already there. It is more like the modality. How do you actually do it? So CNE and reporters actually came and made this article this year with us in the field. They went with the whole team, and you can read it. It's the first time they actually featured an Indian woman on the cover of CNE. And she belongs to one of these tribal belts in Chhattisgarh, in Raipur district, where the sickle prevalence is so high. It happens across all tribals. India has the second largest number of sickle patients in the world. These patients, incidentally, have a very high fetal hemoglobin level. So the Casgevy, which has been approved abroad, which upregulates fetal hemoglobin, might or might not work in the Indian setting. So we need a solution where we can correct the genetic mutation by itself. Okay, and that's when we started working on that. We did all this work. We corrected the genetic mutation back to normal, close to 80 to 90%. We did all the necessary mouse studies, off-target studies, differentiation, patient cells, etc. And we convinced the government somewhere around 2022 that, look, if you have to do a clinical trial for tribal poor patients in India, no company will come and do that themselves. There is no return of investment. Who is going to pay for that drug? Even if it's not costing 20 crores and it costs 1 crore, nobody is going to pay for that. So in a very bold move, the government came and said that we are going to sponsor this. We are going to do a clinical trial for the first time across the world. I'm telling you, this example is not there, where they said that we are going to do a gene therapy clinical trial in India, entirely government sponsored. And there will be all these kind of partners who are there, ICMR, DST, DVT, CDISCO, DCGI, whatever. And as you can understand, as a scientist from IGIV, to bring all the secretaries to the same table, sit and talk to them is way beyond my pay grade. So therefore, Niti Aayog came in and said that we will do that job for you. So you just have to come and ensure that you have the process running. So Niti Aayog came and handled, and that's what they're doing right now. So we are handholding this mission and said that look, we are going to keep all these people, all the stakeholders together, and ensure that we now have a complete CGMP facility made in IGIV, a state-of-the-art CGMP facility for gene therapy, where we are currently doing the scale-up and production. and BMP ward at All India Institute of Medical Sciences, who are a clinical partner. And this is the name of the product, which is Birsa 101. I think you know why Birsa 101. Birsa is, of course, Birsa Munda, who is the tribal superhero kind of, and the first product for sickle cell anemia is kind of devoted to them. I have just, of course, all this work is going, and you will know about it more in later months and in the next year or so. I will end by saying that we have heard a lot about how gene sequencing biobanking actually tells us and identifies novel mutations which are causing diseases. And I'm going to give you a small example of such a therapy, which is for a rare neurotherapy which was called FENIB, familial encephalopathy with neurosurgical inclusion bodies. About 2 years back, 2.5 years back, there was a couple who came from Gurgaon to us and said that, look, our kid is actually suffering from this rarest of rare disorder, where there is a mutation in the SERPINE gene, which causes this kind of a mutation, and due to which she is under continuous seizures for the last 6-7 years. She cannot see, she cannot walk, she cannot speak, basically a vegetable. And we have been funding the New York University Langone Center for Schizophrenia for the last 3 years, and they say that, okay, we can develop a gene therapy for her, but it will take almost 4 to 5 years, and FDA will not give an approval for that even in that period of time. So they reached out to see if we can do something for that here in India. And as you can see, this happens because there is a aggregation of neuroserpin proteins in the neurons. And in a very naive manner, we took up this case. If I had a chance, I would not do that again. But essentially, I thought that, okay, there is a patient who has to be treated, and what would entail to do that treatment was to make fresh gene editing proteins which would actually correct this mutation back to normal using base editors, whole genome sequencing, digenome sequencing to see whether there are any kind of off targets. We only found one single off target across the genome, which was fortunately for us lying in a kind of a gene desert. We made iPSCs from that patient. We converted them into neurons. We saw that upon the gene correction, the iPSCs now had reduced neuroaggregation. So phenotypically, it would produce the effect that we kind of desire. Now, all that was fine. The CRISPR was made. You still have to do that injection into the brain and correct that, right? So we needed a vector, and the only vector that actually works in brain diseases still for gene therapy is AAVs, adeno-associated viruses. So I had a collaboration already ongoing with a very dear friend and collaborator at Narayan Nitra, Orko Shubhu Ghosh. He has a facility, a GMP facility, where you can actually make AAVs. So together we work together to make these, dual AAV vectors for adenine base editors, CRISPR editors, corrected the mutation, did all the necessary mouse studies, close to 200, 300 studies, animal studies, where you find what is the route to the brain, intrathecal, intracerebral, ventricular, or something else to find out what is the best route, and then assemble a team of kind of very ambitious doctors who said that, oh, this is routine for us. We're going to inject this kind of, for scientists like this, to even think that this is routine is a big thing, that you could inject that into the brain just by a dose into the cranium. At this point, I'll just give you a bit of a timeline how this all worked. It started in 2022, and throughout this process, we received unsurpassable help. Basically, we had Oren Devinsky, who's the head of the NYU, who kept on helping us with how to develop this kind of therapies. Dr. Miranda, who is in University of Rome, who happens to be the only person studying this rarest of rare neuropathy. So she had antibodies which she shipped within 72 hours just because there was a patient online that has to be saved. Somewhere in, Orko started making these batches of GMP sometime in August 2023. We started approaching the regulators, DCGI in this case, in Drug Controller General of India, explained to them and received tremendous support for them. They said, look, this will fall under a sunshine trial or a N of 1 trial where a patient has to be saved. We will not ask you to go and do your monkey studies or chimpanzee studies, just a basic set of studies that is required to make this therapy go into a person and save them. So they came for audit, they checked all the facilities, all this was done. And tentatively around December, January, we expected that maybe we could start getting this team of doctors ready to do the injection. But unfortunately, in October, the patient passed, even before we could actually give this therapy to her. She's passed away from some complications, respiratory complications. She was already drooling when I met her in August, and she had to be, brought. And it was a very moving episode because, Uditi, this is the patient actually. The parents were, she could not see us, but the parents were actually telling her that, look, here we have the doctors who are going to treat you, who are going to make you well. But that did not work. You can read more about this in this article in Nature, which Nature did last year. They covered this whole story. What I wanted to emphasize here was its tremendous amount of dedication by the parents who actually put all their eggs in the basket of science, not in the basket of anything else. Okay, and that sets a course which is almost unprecedented. And there has been numerous, how to say, reaching out of parent groups and so many of them after this episode where people know that such kind of therapies are there on the horizon, but it takes an ecosystem to really make sure that they reach the patient. I'll skip this. We'll just go to the last slide. Yeah. So just want to emphasize that there is a very long queue of patients, and time is a commodity for most of them. So I talked. Yeah. So we can make our, how to say, the CRISPR editors as safe and possible. But unless you actually go and treat patients, there is no other way you will know whether the therapy works. You have to just stop and go to the patient. And of course, pricing, you can solve this pricing issue a lot with endogenous production, indigenization of technologies, which comes when you have an IP. Just the fact that there is an American patent means that now we have industries, big industries, small industries, startups, everybody licensing, coming ahead and making the CRISPR products in India now for multiple disorders. I can tell you already about this, for multiple disorders and indications, this has started. And of course, I talked about awareness. There is no other way of awareness than actually patients themselves coming and awareing. This is our Chief Justice D.Y. Chandrachud. He invited us to his residence, and that's when I came to know that his two daughters are suffering from Nemaline muscular dystrophy, and they wanted a gene therapy cure as well. And it doesn't differentiate, the disease doesn't differentiate whether you are the daughter or the son of a Chief Justice or you that of a very, very poor person. So that tells you that it's an ecosystem which has to work towards creating that. Starts from funding, ends with the doctors, and patients are the most important factor in this whole thing. So when I come and speak to you, this is the most important slide actually. When I come and speak here, I'm just doing the selling stuff. I'm just telling you a story. But the actual work is done by a large number of people, students, collaborators, clinicians, across the world, multiple people who are working, and we are of course very generously funded as well. So thank you to the audience who is there to listen, and if you have any questions, I'll be happy.
Thank you very much, Doctor. I think it's a wonderful, very interesting, insightful information in your talk. Due to time limit, yes, we may allow only one question now.
Just a comment to make. Congratulations for the path-breaking work. It's really illuminating, and I think it's time for haplobanking to take off, where haplobanking would set a platform, the go-to platform for HCST transfer, and then use it as a model for hemoglobinopathies in India. So your take on that?
I think what you're saying echoes with a lot of clinicians whom I'm discussing with. Of course, that's comes with a lot of cost and an infrastructure development. If I know correctly, there are only 105 BMT centers in the country of so many people, million people. Even if we have this gene therapy successful, how do you actually manage to give them to so many people? Because it ends with a BMT, right? We will make the product, but it still has to go through a BMT. So the infrastructure is still required to even do this kind of a banking facility. I think it's a very pertinent thing. As more and more gene therapies will come, a lot of them through HCST, this issue also has to be raised in the right domain. I think NHS or even the national, the Department of Health has to be involved. I agree with you.
Thank you, Dr. Devajyoti. I would request him to stay back on guys, please, and request the chair session because he has to leave. So kindly felicitate him before he leaves. we may resume the session further for 2 more speakers.
Yeah, it's time to move to the next speaker. Our next speaker is Dr. Priyank Shukla. He's a senior lecturer in bioinformatics at Ulster University with a background in biotechnology and bioinformatics. He completed his bachelor's from MJPR University and master's from CSGM University in India, and did his doctorate in bioinformatics from University of Bologna, Italy. His research focuses on developing AI-based methods for biomarker discovery and patient stratification in inflammatory diseases, cancer, and multimorbidity. He has held postdoctoral positions in Austria working on NGS projects related to cancer inflammation. And Dr. Shukla also leads the personalized medicine program at Ulster University and has secured significant research funding from major UK organizations. His teaching interests include biocomputing, biomedical informatics, applied in bioinformatics, and he has received several awards for excellence in teaching and research. Over to you, Dr. Priyank.
So good afternoon, everyone. First of all, thanks to Virendra here who invited me for this. It's an absolute pleasure and honor to be here. And thanks to the chairs for the introduction. And yeah, so I come from Ulster University School of Medicine. There is a center there called Personalized Medicine Center. It's been like 10 years. And we work in several disease areas, and one of the things what we do is also biobanking, where we have been recruiting patients across several disease areas. And it's around like 5,000 patients, which may not be a big number, but in the context of UK, specifically in the context of Northern Ireland, it is a big number. And my interest over there has been applying machine learning and AI-based methods for developing early diagnosis, prognosis, and response to treatment sort of algorithms, and also in multimorbidity, which I must say, like last 4 or 5 years, I've been looking into it. And right, so what is multimorbidity in context in terms of background? So it refers to more than one condition, and I must confess it's still a very vague term. So anyone who is having 2 or more disease is categorized as multimorbid. But that doesn't really help to my understanding in terms of developing any treatment, because for developing any treatment pathway, you need to properly characterize the disease. But in this context, there are so many diseases. How do you do that? And that sort of brings me thinking that, okay, is there any way we can further characterize them, further sort of stratify them into smaller subgroups, which, if we can look them in further detail, might help us to develop some personalized medicine treatments. Now, this could be any diseases ranging from non-communicable mental health infectious disease. And we have been listening around the term UK Biobank several times, hopefully since yesterday. And that's what I'm going to present today, that we have been looking into UK Biobank data, trying to analyze. So what exactly is the problem? So it is really emerged as a major global public health
issue, and a lot of adverse health outcomes, example, disability, poor quality of life, mortality, and so on. And especially older people more than 40 years of age do get into these issues. Now, so what we did, like around 2019, we went to UK Biobank and applied for a project where we wanted to look into these 500,000 patients data from the angle of multimorbidity and all genomics, proteomics, everything, whatever was available to analyze it. But when it came to really analyzing this, and I kind of believe bigger problems sometimes have simpler solutions. So all what we looked into it was just disease diagnosis, nothing else. So in that UK Biobank, which is such a rich repository of lot of data available, the only thing what we pulled out in terms of variable was what is the disease diagnosis someone has been reported. Using that data, using that information, we pulled out something called Charlson Comorbidity Index. It is a parameter to measure how severe multimorbidity is. And then all what we did was a multiple correspondence analysis to see if there is any cluster happening. So that's basically the pipeline, which involves everything. I can see it's a bit blurred. I don't know why, but it looks very nicely on the computer. But I'm more than happy to explain what it is. So like I said, started from 500,000 patients, filter down anyone who has been diagnosed with 2 or more diseases. That was around 70,000 patients. And then pulling out specifically simply in ones and zeros, binary number, what all diseases someone has got, categorized into 17 chapters of ICD-10 coding system. Plugging that data into an MCA analysis to visualize if there is any clustering happening. And we found 5 clusters, and I will show you later. Then, as for the pipeline, trying to explain you further what happened next. Once we got those 5 clusters, we thought, okay, is it really happening, or is there something wrong with the data? So, and when I say something wrong, I don't mean that people have reported wrong, but is it just a number game of the multiple correspondence analysis, which is pulling out these people into 5 clusters? Or is it really something there that people could be stratified into 5 endotypes of multimorbidity? So we said, okay, let's pull out these groups into 5 clusters. Let's separate them out and try to train a model to see whether the model is able to predict into the right categories what the MC analysis is showing us. So basically validating a computational experiment via another computational experiment. And to our results, close to 90%, the model was giving the same results what an MC analysis was. So this is basically the whole pipeline, what I tried to explain in a nutshell. But let's go to the results. So what we saw was this. So left-hand side is an MCA plot. Each dot is a patient. And as you can see, nice 5 clusters over there. And we thought, okay, it looks beautiful, but that's fine. But what is exactly driving these clusters? We went on to investigating the variables which are basically pulling these 5 clusters apart. And like I said, these were broad diseases which are used in the Charlson Comorbidity Index to measure the Charlson Comorbidity Index. So, long story short, it's basically paralysis, stroke, and dementia in the UK population, which is driving or which is pulling them apart into 5 different endotypes of multimorbidity. And we said, okay, let's see further what that would mean. So we went on further looking into other variables. Like I said, we initially started with just diagnosis, but then we thought, okay, let's see what are the other things which are different in these 5 endotypes. So first thing was proportion of how the 2 genders, male and females, are grouped into 5 clusters. So we see that the proportion of females is going down as the Charles-Cromartie index is increasing. So basically from 1 to 5, if you see plot number C, the CCI, the Charles-Cromartie index is increasing. And if you now compare it back to A, the proportion of females is going down. So basically, the males have much more multimorbidity. And I kind of feel maybe it relates to the age thing as well, that the age of at death, which is plot number B here, it was also increasing as the multimorbidity or the CCI was increasing. Now you might argue that if the Charles-Cromartie index is increasing and the severity, people are much more in a problematic state if they are into group number 5, then how come they are actually living longer in terms of the age of death is much longer. So we kind of hypothesized, I mean, we don't know what could be the reason, but one of the reasons what we thought could be the people who are really bedridden. And when you're bedridden, you are much more taken care of as compared to when you are still walking around with your problems. Again, this is just hypothesis. We don't know. We need to look into it. The other interesting variable we wanted to look was this, if I have to talk more simple words, is poverty index. So basically, the UK Biobank also has the information about every patient in terms of how what exactly grouping they come from in terms of the poverty. And again, like we see, there is an increasing trend that people who have more multimorbidity tend to have much coming from a more poor background. So again, this could be linked to the fact that to the education as well, that people who have money, who have more education, they are better informed and they take care of their children. But something which is not new, I must say, it is already being known that people coming from a poorer background have more multimorbidity. But that was like basic interest in terms of, let's see how those 5 endotypes characterize. But then we wanted to really investigate further in terms of coming back to those diseases. And we found 3 paralysis, stroke, and I think it was dementia, paralysis, stroke, and dementia. Those 3 diseases, what's happening with them? So what you see in figure number A here is that we have the 19 diseases which are used in the Charles-Cromartie index calculation, and then we try to look into the basic distribution, that how many people in each cluster have those diseases. And as you could clearly see out, it's like stroke, paralysis, and dementia. These are the one of the big diseases over there, but also they are very, very different when you compare across the cluster. And then we thought, okay, that's the distribution, but what is how they are related, meaning how often do both the diseases, more than one disease, come together. So on the bottom you see 5 heat maps showing you that in case of cluster number 1, it's basically the pulmonary group, the pulmonary diseases, which are very, very common. Then I think in the second one it is stroke, third one stroke and dementia, fourth one. But this heat map gets much more clearer when we thought, okay, let's try to build a network of what's really happening here. So as you can see, cluster number 1, even though it's multimorbid, but it's kind of spread across several diseases, and it's mainly pulmonary, which is coming out. Number 2 is stroke. Number 3 is stroke with dementia, which you can see quite commonly happening. But 3rd and 4th, which are much more severe cases, you have stroke and paralysis, which kind of is obvious, I kind of feel, are much more correlated. By the way, each node is a disease. The size of the node tells how much prevalent that particular disease is in that cluster. The thickness of the connections tell us how often that those 2 diseases are present in that particular endotype. And the final cluster, as you can see, the stroke, dementia, interestingly, and paralysis, these 3 are connected together. So basically, a good number of patients over there, they have those 3 diseases as very common occurrence. So to be honest, that was the first step for us. In simple terms, that multimorbidity, which is very vague, can we find something? Can we sort of divide that multimorbidity nomenclature into 5 types or something or not? So in a way, we kind of claiming that there are 5 endotypes of multimorbidity, and we went on publishing this in Briefings in Bioinformatics, which is one of the good journals in our field. And this work was done by the PhD student from a lab called Mohanty Prasad. Very, very intelligent man, I must say. Product from JNU, so anyone sitting from here should give a shout out for him. He did his master's from here, came to me for doing his PhD, and he got vice chancellor researcher studentship over there to do his PhD, and now he has moved on to, which is not coming on to the slide, he's moved on to Glasgow for doing his, he's a postdoc over there, technically a bioinformatician, but excellent guy, and this study was funded by the Academy of Medical Sciences and Public Health Agency, and also European Union funds and Department of Education. Now, this story doesn't stop here, just 1 or 2 more slides. Because we found something interesting in UK population, we thought, okay, this is maybe specific to UK. What about other ethnic groups? What about other population around the world? So we went on establishing an international consortium of multimorbidity research and development, which we got through a Global Challenge Networking grant from the Academy of Medical Sciences. And we got roughly 9 to 10 countries on board, especially from South Asia and Southeast Asia. And we had our first meeting in IT Monday last year. And the idea there is basically just to replicate what we did in the UK Biobank to establish if the term multimorbidity, which is so much important these days, given the population is aging across the world, are we going to just be happy with calling it multimorbidity as a vague term, or do we really want to characterize it properly and establish which population in the world have which types of endotypes of multimorbidity? So that's basically the aim of this consortium. We are working on it to move this forward to get some more money and recruit patients from these parts of the world. Not really recruit, I mean, the data is already there. Most of the hospital records do have diagnosis. So the idea is basically instead of recruiting patients, pull out the data and run the same pipeline and see if you see exactly those 5 endotypes or they are different. So with that, I would just thank everyone from the team and the funders and you for listening.
Yeah, thank you, Priyank, for this exciting talk. We would request any one question from the audience.
Very interesting study. Paralysis is a manifestation of stroke. Is there a reason why you separate?
Well, I mean, we didn't do, I mean, like I said, I don't come from a disease background. So for me, that wasn't really something I was purposely looking into it. But we were following Charlson Comorbidity Index. And the way it is calculated are those specific diseases. So Charlson, the scientist, says that these 18, 19 diseases, we need to check in a patient. And then there is a mathematical formula to calculate what is the value. The value will then say whether you have high multimorbidity or a low multimorbidity. So he basically said paralysis to two separate things. But I do agree from the clinical point of view, we'll have a different view on that. But like I said, I mean, we were not really getting into that.
Okay. Thank you, Dr. Priyank, for your talk.
So now our next speaker is Dr. Mohanty Bhatt, Assistant Professor, MMI HCST, Nepal. Dr. Mohanty Bhatt is a distinguished academic in the field of clinical biochemistry and laboratory medicines. He currently serves as an associate professor at Manohan Memorial Institute of Health Sciences in Kathmandu, Nepal. Dr. Bhatt completed his Bachelor of Medical Laboratory Science in 2008, followed by Master of Science in Medical Biochemistry in 2010. He earned his PhD in biochemistry from Congo, Nepal University in South Korea in 2013. His academic career further includes a postdoctoral research position in Kongo National University Hospital, South Korea, where he worked from 2013 to 14. Dr. Mahendra Prasad Bhatt's research interests are primarily focused on areas within clinical biochemistry and laboratory medicines. Dr. Bhatt has significant research profile over 25 publications that explore areas such as hematological parameters in chronic kidney disease, protein estimation methods, and clinical laboratories, and cardiovascular disease risk assessment in populations with thyroid disorder. His work has been widely cited, reflecting its impact on medical and scientific communities. Dr. Bhatt is also an active member of Nepal Health Research Council and continues to contribute research and teaching in his field. His work in these areas has contributed to a better understanding of disease biomarker and clinical diagnosis process, which can aid in improving patient outcome. So welcome, Dr. Bhatt.
Thank you so much, Chair, for very long introduction, even though I have many significant contribution in various scientific areas. Anyway, these are not the point for discussion here in today's presentation. So I'm just here for talking about the future, future emerging future biobank concept. We can discuss here some proposed, modality, how we can go with the biobank for the future health security. So anyway, thank you so much. Thank you so much, BBIF, for inviting me from Nepal. However, I'm working with BBIF since long time as a member from the nearby country. Today's talk is about an emerging biobank as an emerging component of global health security. We say many things about the biobank, like biobanks are very important for the treatment purpose. Biobanks are important for the research purpose. Research and development can be, improved in future for the health security. But let's go beyond the practice. We have to go beyond the practice. We have to go for the health security for the next generation. So I'll go through the background, next generation human security up to height and depth, from the Big Bang to the evolution of planetary system as Earth become become like a livelihood. It's like nowadays we're living together. So if we see the past and if we see the future, there are many health threats for the human race. That's why here is now time to think about the biobanking that can contribute the future of health safety in the universe. So universal health coverage is the one of the very important part of UNDP as well as WHO is working on it in every country. So as it is constituted in 1948 WHO, 1978 Alma-Ata Declaration proposed this universal health coverage. in which our biobank components are already active. However, these are not still, synchronized and standardized to make the scientific, outcome. So even though health insurance are, applied in various countries, so these are also insecure. These are also for insecure in the universe of safety. So security issues are discussed in 1994 when UNDP put forward a document, Human Development Report in 1994. Amartya Sen was leading from the India at that time. They put forward economic security is very important for the global security, food security, health security, environmental security, personal security as well, community security, and political security as the major component they put forward for the future. Here, Global Health Security Agenda 2024 from this state. So there are 67 countries collaborative action put forward in exactly 2018, I think. So this agenda is collaborative agenda of 67 countries working toward the health security for the future. So in which various, health security agenda were implemented in various countries by the CDC, by the lead of CDC. So this is one of the health security agenda put forwarded by the UNDP. So for that, Global Health Security Agenda 2024 vision of world safe and secure from global health threats posed by the infectious disease, whether natural, deliberate, or accidental. This is the before pandemic of the COVID. These strategic, plans are well developed. So the mission of this GHSA 2024 was to realize its vision. This will facilitate the effective integration of the multi-sectorial and multi-stakeholder work to achieve sustainable and measurable result toward common target for the global health security. Core principles were country ownership, inclusiveness, cost effectiveness, mutual accountability, transparency, multi-sectorial approach, measurable progress and impact, sustainability, partnership, and proactivity were the component of that. So global health security agenda now I'm coming in the point forwarded statement for the laboratory system where biobank and objectives are linked here. Now, important component of global health security is the laboratory diagnostic part, real-time biosurveillance of the disease through a robust national laboratory system, effective modern point of care and laboratory-based diagnosis and research is vital to timely addressing of global health security issues stated in the document. Step to ensure health security, we can go through the nationwide national health security should be assured through the multi-sectorial emergency preparedness. However, all the countries are doing the emergency preparedness plan. However, the plans are still not integrated and collaborated properly in the globe. So we must go the more integrative and collaborative approach. So sustainable development, health professionalism can be developed, resilience, quality health service, and quality health professional education are the major component of national health security worldwide, so that we can go more scientifically and concisely to compare the upcoming health threat in future. So to ensure national health security, we must go collaboratively through the Ministry of Health in collaboration with Ministry of Defense, Home Affairs, as well as private sector collaboration. NGO can be the major part of that health security agenda of the country. Health emergency preparedness, we have the very good example of safe blood and blood component for our blood banking systems are well equipped in the country to support the emergency, therapeutic cell plasma antibody molecules can be utilized from the blood of the donor. Similarly, we can make the biobank set up like safe biobanks for all is very important. This is the need of time and beyond we can plan like this. So these are obviously important for the research, genomic effect of health on the health, transcriptomic effect on health, proteomic effect on health can be beneficial in the future. In therapeutics also, organ transplantation, tissue transplantation, cell-based therapy can be developed, gene therapy, proteomic therapy, and prophylactic, vaccine production can be integrated with the biobank system in future. So there are major contributor promoting the health security worldwide. UNDP obviously initiated as well as promoted the health security in which biobank can play an important role in future. Here, FOA, WHO, OIE, and World Bank also are contributing and encouraging the health security of the future. Role of biobanks in future, we have to focus on health system development strategy towards the stratified health in future, like a personalized health system can be developed. These personalized health, stratified health system can be established only if we develop the biobank and biobank support to the all health care delivery system in the world. So WHO have a very important One Health approach in which also there is very important role of biobank. That's why we can go forward to improve the biobank system, which can integrate the objective of the WHO goal. So in cooperation with the lifelong learning, health professional education development is very important, in which we can develop the curriculum for the health security purpose, in which one of the components can be the biobank system setup as well as implementation for the services. Professional competency toward the health security is very important for the future. Curriculum upgrade, biobanking, next generation AI system which can integrate the biobank worldwide. So develop attitude and stratified health services. So attitude of stratified health service is very important. That can be created only if we plan and develop the curriculum like personalized medicine is important for future health. So PBL system CME can be upgraded for the future. Evidence-based clinical practices are very important. R&D are obviously important, in which everywhere biobank can support the development of all those components. So conservation of natural products are still very essential, because natural food, natural plant resources are nowadays damaged by the environmental threats. So for the human security, environmental friendly industrial development, heritage and natural conservation of the energy, upgrade the level of the civilization, approach extraterrestrial life shifting the future challenge for the health security. That's why biobanking are also major player in this field. So Prevention and control of disease, obviously biobanks play a very important role. Infectious outbreak, metabolic disorders, cardiovascular diseases, cancer, signs of infertility, genetic disorders, aging, everywhere we can integrate the role of biobank in future, so that biobank must go more scientifically, become more open access to the society, to the country, to the professionals. So open access strategy for health research through biobank can be made through research collaboration of health security globally. National guidelines on networking biobank must be, standardized worldwide. National guideline on networking biobank is also very important. A common ethical approach should be there. Sharing biomaterials is very important. To share the biomaterial, there are very, racial as well as some hurdles are there that must be, resolved. Sharing data is one of the, again, more big challenge that must be more friendly and scientific, so that we can assure the, the data sharing process. So sharing digital health technology is again important in the future. So this is one of the insight I put forward on behalf of BBIF, insight into the biobank networking. Biobank can be can network one of the government or non-profit organization can lead, like BBIF can make a network of biobank, including non-human and human component in the biobank. Pathogen, animal and plants can be included in non-human sector. I am very surprising and happy to see, buffalo biobank, tissue biobanking are developed in India. Everywhere, every component, small biobank components are already developed. Just we have to synchronize and we have to make the standard networking of the biobanking system. For that, major contributor can be the established biobank, like UK biobanks can suggest and can support the new biobank development in the other countries, so that they can lead the networking of biobank, and we can use the single platform from even though Nepal, from India, from UK. That doesn't matter. We should break the boundaries of hurdles in the biobanking system. So in this way, human biobanking systems are again very big, broad. Human organ can be stored and shared. Human tissue, cells, normal, abnormal, those can be utilized for the preventive as well as curative services, utilized for the research and development, utilized for the ensuring health security for the future. So this is one of the proposed classification system to categorize the biobanks based on the application in the human health security. Essential collaboration can be global. That global collaboration can, network the national biobank systems, and national biobank system can network the multi-sectorial and multidisciplinary approach and multidisciplinary biobanks together, and we can make a single biobank. I heard many type of biobank, but biobank or single biobank cannot store all those systems in a lab. So I have a very bad experience. I'm working in a Nepal Health Research Council. They had planned during the COVID, during COVID, many, health sector who have the fund, enough fund, they want to put fund in the investment. So they invested huge money in the storage system, making storage system is a biobank. However, they could not succeed to run this biobank in Nepal. So That's why now we are planning, we must first collaborate, we must first get the support, make the guidelines and act for the biobank so that we can utilize those biobank materials, whatever we stored in a single biobank. So Nepal Health Research Council cannot store the whole biobank system in a single lab. That's why this biobank is one of the entity, can be or national entity or global entity. If we become in a network, that can be a single entity. That could be a future, best option for the biobank. So joint collaboration is very important. Global biobank database here I'm talking about. Biobank is not only supporting the sample sharing. Biobank is again important for the global data sharing. Data sharing act and data sharing, guidelines are very important to be developed. That's why I request BBIF to make some guidelines, to insist some guidelines to develop this type of data sharing system in India so that we can learn from India also. So thank you so much. I had no data from the research here. I just want to put some proposed, plans to develop future of biobanks. So new ideas are welcome to contribute proposed BBIF classification of biobanks so that we can make a small, publication. Multidisciplinary, multi-sectorial approaches for the health and prosperity is the main agenda of the global health security, in which biobank is becoming, it must become the major.
Yeah, thank you, Dr. Mahendra. We look forward to enlightening collaborations between Nepal and BBIF in future. Yeah.
Thank you, Mahendra. Thank you for that one. BBIF is already started the South and Southeast Asian Forum. We are in that one. We are going to have meetings soon. And as you see that BBIF has already started the multi-sectorial collaboration. We have, even in this conference, there is a one health organization is collaborating. That one. So, including the South and Southeast Asian countries, we are going for the not only the biobanking but also the one health. So that we can not include only humans, but we are going to include all sectors which have bio samples, either that is environmental biobank, animal biobank, human biobank.
इसका नाम लो
Thank you so much.
वही कल जो बाइट्स बनाए तो वही ना जो बाइट्स बनाए तो वही ना कल जो बाइट्स बनाए.
Thank you, Dr. Mahendra. Dr. Mahendra, kindly stay back. I would request our chairpersons to verify the speakers of the session. Dr. Anita Mahadevan. It's always an honor for us. Dr. Priyank Shukla. Now I request Dr. Anita to kindly come and dais to felicitate our chairpersons who had been very patient for this session. So very soon we'll have our lunch break. Thank you all. We'll rejoin our session, that is workshop after this, after post-lunch. Lunch is for half an hour, if it permits. Half an hour we'll keep. So we'll try our best to come as soon as possible. Thank you all.
अरे यार क्या हुआ चलता है नहीं